Hello, and thank you very much for attending. It's really a pleasure to give this talk from across the other side of the Atlantic Ocean. I'm going to be talking about some recent work that we've done on smallpox and give you some general introduction and background and a bit of history as to what the virus is and what it did and what it may do in the future and so on. Here's a little bit of an overview of what I will discuss. I'll give you a bit of history. If I remember, I will give you a warning and alert you that I'm going to show some images of people who had smallpox. Hopefully you'll have time to close your eyes if you don't want to see them. I'll talk a little bit about ancient DNA because the work that we've done has involved getting sequences from the virus out of skeletons from people who died about 1500 years ago or so. I'll tell you a little bit about our results, a little bit about immunology, about how viruses get into humans from animals, a little bit about vaccination, and something about the future, perhaps. On the left there is one of the skeletons that we got DNA from. Usually we get it from the teeth, and sometimes from a bone in the inner ear. And that's called VK533. That was the name of the sample, the number of the sample. In fact, you can actually see, let me just have a look here. I'm having a bit of trouble with my presentation. There we go. You can see just on the person's right shoulder, there was a dagger that was in the grave. That's a grave good that sometimes Vikings were buried with things like jewelry or weapons and so on. Okay, so a little bit about history. I think people probably in general are not very aware of viruses. Even though we have a pandemic going on, there's not a lot of understanding about viruses, and they're actually really quite amazing. And one of the things which is amazing is how old they are. The viruses have been around basically forever, possibly since before there were cells and before there was DNA and sort of before anything. So we know that some viruses are certainly hundreds of millions of years old, and they infect everything. You know, they infect bacteria, they infect basically everything. They maybe even infect other viruses. And pox viruses specifically, which is what we're talking about today, they infect all kinds of things like insects, birds, reptiles, and mammals. And in particular, we're talking about smallpox, which is one particular pox virus in humans. It only infects humans. And we don't know how it got into humans or when exactly. But we have some clues from up to about, let's say, 3,000 years ago. It's hard to know because the records from, let's say, 1000 BC, they're quite unclear. The written medical records, they're quite ambiguous. Things could have been measles, they could have been chickenpox, they could have been smallpox, they could have been something else. It's also possible that the way these diseases affected humans has changed over the millennia, and things that had a certain behavior back then don't do that anymore, or vice versa. Another thing that's interesting, perhaps to a US audience, is that George Washington and Abraham Lincoln were both infected by smallpox. They're not the only presidents who have had viruses in the pandemic. As you know, there's a recent case. Also, Woodrow Wilson was infected by smallpox in April of 1919. Anyway, I've got a little excerpt over here on the left from a very good book that gives you some sort of clues about the importance of smallpox in history and its effect on everything from kings and queens and so on down. And the author is a man called Donald Hopkins, who I think is based out of Baltimore at the moment. Anyway, he says that the outbreak, the disease embarked on a deadly series of attacks, and it's a regicidal rampage without parallel anywhere else or at any other time. It killed a queen of England, an Austrian emperor, a king of Spain, a czar of Russia, a queen of Sweden, and a king of France in the 80 years before 1775. So you can see smallpox had a pretty big impact on Europe in the 18th century, and of course after that as well. All right, so I'm going to show you some images now. If you don't want to see them, close your eyes, and in 4 slides time, I'll tell you to open them again. But I would urge you to have a look, because they're not that horrific, but they will perhaps take your breath away. And there's an important lesson here that I think is worth appreciating visually. Okay, here we go. Oh, here we go. The first one here, there's 2 boys. They look like they're about 12 years old. This is a photo taken in England in the early 1900s. On the left, you have a boy who was not vaccinated against, well, he was not inoculated against smallpox. And on the right, you have a boy who was. I should say vaccinated, that's the correct word. So you can see the contrast. They both got smallpox, but the one who was vaccinated had a relatively good outcome, and the one who was not vaccinated had a really horrific outcome. In the next slide, you can see a man who has smallpox. Obviously, it's just his face, but that's a pretty full-on disease. This is an image from Bangladesh in 1973. This comes directly from Wikipedia. And the final one is another patient suffering from smallpox. And you can see the extent of what the disease does to a person. Okay, I've skipped the horrific images. I hope the majority of you are actually watching. So you can appreciate really what smallpox did to people and how horrific and terrifying the disease was. Nothing at all like what the COVID-19 disease is doing to people, where many people have quite mild symptoms. Some people wind up in hospital, but not very many. Okay, so what we've been doing in our research is looking at the DNA from humans who died up to about 30,000 years ago. And it turns out that you can get the teeth, or get a bone, which is illustrated in the bottom picture there, from the inner ear, which is a very dense bone called the petrous bone. And if you process these correctly in the lab and you extract, you grind them up and so on, you can extract DNA. And the DNA is from the human, from all of those years ago, which is kind of incredible. Most of this work was done to try and look at the human genome. But it turns out that if you do this in the right way, you also wind up with DNA from other organisms. You may get stuff from a parasite, you may get contamination from the thousands of years in between, but you may also get things like bacteria and viruses that infected the person and that they died with and maybe died of. And so a lot of examples have been found, for example, of people who died of the plague, which is a bacteria. But we also get viruses, and in particular, we found smallpox DNA in the teeth of Vikings, or people who died in the Viking Age, from around about, a little bit earlier than the Viking Age, from around about 600 to about 1000 AD. And you can pull out this DNA, you can put it back together to get the sequence of the virus. You're not actually getting the virus itself. The virus is gone, but you can still get the DNA sequence. And we did that. We found about 26 people who had smallpox infections when they died. We don't know if they died from smallpox, but we know that they died with a smallpox infection at the time. It may well have killed them, but we can't know for sure. Anyway, here are 13 of them where we got a lot of the smallpox genome from the virus out of the DNA from the teeth, or the petrous bone. And you can see that the ages are quite variable. Some are male, some are female, obviously. Most of them are from Scandinavia, and you can see the time period. As I said, it dates from around about 600 to around about 1050 for the old samples, and then there's a couple that are in the 19th century. And here's a little map showing you where these things were located, and in other samples that we looked at that didn't have smallpox in the grey dots. Because I'm Australian and I'm allowed to make fun of people, because that's my cultural background, I put a little map of the world there for you, just so you know where Scandinavia is, in case you didn't already know. Anyway, and we found one sample at the University of Oxford, which was a cause of much celebration because we were at Cambridge at the time, and we found one in a mass grave. Actually, it's quite interesting that there was a king from the year, I think, 1003 called Ethelred the Unready, who decided that all Danish people, or all Vikings, let's say, in what was then, England and what we now call the United Kingdom, should be executed. And they were killed, and one of them had smallpox. Okay, so what did we find? This is where we get into the science, and I don't want to go into detail because it will take too long. But the thing which is most interesting and most relevant here is that the virus sequences that we found in the old smallpox are really different from the sequences that are found in the modern smallpox. And viruses, just like other organisms, they have genes. Humans have got genes, bacteria have got genes, animals have got genes, and so on. And the genes affect behavior. They affect, what proteins get made and what things are possible, and so on. And the smallpox virus has, let's say, about 200 genes, and some of them have a function which is known, and some have functions that are not known. And we found a difference in a lot of them. And in fact, in the 4 sequences that are the most complete of the ones from our study, there was a lot of diversity within those 4 sequences. And this illustration, the colors in the columns are showing you differences in the different genes. And along the bottom, you can see the names of the genes. They won't mean anything to you. In fact, they don't mean anything to me either. And on the left, you can see a little tree, which is sort of like an organizational tree of some of these viruses. I'm going to show you a little bit more detail, so I'll move on to the next slide. But the most interesting thing to take away from this is that there's a lot of diversity in the old viruses that is not present today. Well, it wasn't present in the 20th century when smallpox was eliminated. This is what's called a phylogenetic tree, which is, it's not a real tree, but it's a tree showing the relationship of collections of sequences. And up here on the top right, we have what we call the modern sequences. These are things mainly from the 20th century, and there's a few that are a bit older, from about 300 or 350 years ago. And they form a group because the sequences are very similar, which is to say the DNA is very similar. And then we have a second group, which is the ancient sequences that we found that go back about 1,000 years earlier than the previous oldest sequence. And you can see that these fall into a very different area of the tree. They're on their own branch, if you like, and they split into 2 halves because, as I said, there's some diversity within these ancient sequences. And then down the bottom here, I've put a circle around the other closest pox viruses. One is the camel pox virus, and the other one is called tetanus, which is found in gerbils. There's many other pox viruses, including one that I'll return to soon, which is called monkeypox, which is actually quite a concerning pox virus. All right. So people often say to us, you found these ancient viruses, these virus sequences. What do they do? What's the meaning of this difference, and why were these things so different, and why did they go extinct, and what did they do? How did they affect the humans? And the answer is that we just don't know. And the reason that we don't know is because immunology is extremely complicated. Here's a tweet from Carl Zimmer, who is a science writer at the New York Times. This is from a couple of weeks ago. And he says, the immune system doesn't do simple. And he's got a little image here of some of the bits and pieces that are involved in the immune system reaction to an incoming pathogen, like a virus or a bacteria. And actually, his image is quite incomplete. This thing which he's showing, which is very complicated, is actually just a part of the overall immune system. It actually corresponds to that little green box at the top of this diagram, which is taken from another book. So that thing that Carl Zimmer was pointing out is just one box on this image. And this image is just the top half of this box, which shows you even more of the immune system. So the immune system is incredibly, mind-bogglingly complicated. It's insane. It's probably the most complicated thing I've ever tried to understand, and I didn't get very far. And to go along with that complexity, you have the pox virus. And this is an image of the genome of a pox virus. And all of these colored, blue and green and red little sections corresponds to a gene. So these are the hundreds of genes in the pox virus that interact with the immune system. So there's an extremely complicated interaction going on, and it's very hard to study, or in the case of smallpox, it's impossible to study this because you can't do these experiments because smallpox is so dangerous that the experiments are illegal. There's no way to do them. Smallpox is stored at the CDC and also in Russia, and basically no one is allowed to do these kinds of experiments. So the closest we can get is to look at a different virus, like for example, vaccinia virus, and we look at it in a different host, like a mouse. But at that point, it's no longer smallpox, and you're no longer doing experiments on humans. So your results may not really be valid. Okay, so immunology is complicated, smallpox is complicated, and we don't really know what all this diversity did. Okay, so I'm going to change subjects a little bit and tell you something about zoonoses, which is a fancy word that basically means how do diseases get into humans from animals? And maybe all human viruses, almost certainly every human virus that you've ever heard of comes from an animal. That includes smallpox, which is likely to have come from rodents. It includes the current coronavirus, which has likely originated in bats. It includes the old SARS virus from 2003, which also is a bat virus. It includes the Middle Eastern Respiratory Syndrome, which is another coronavirus. It includes Ebola, HIV, rabies, West Nile virus, measles, influenza, Zika, which was in the news a few years ago. All of these viruses have come from animals. And they get into humans in different ways. Sometimes we kill the animals and we eat the meat. Sometimes we lick their faces. Sometimes a camel spits out half a liter of saliva and it gets into a human. There's lots of ways that this happens. Next subject is vaccination. Edward Jenner, who is pictured on the left here, did an extremely daring experiment in 1796. He actually wasn't the first person to come up with this idea, but he was the guy who really dug into it and followed up on it and wrote about it and made it well known and got all of the fame, probably justly so. Before Jenner came along and tried to vaccinate this young child, there's a process which had been called variolation, which had been going on for a long time, probably 2,000 years, where people realized that they could protect each other and themselves against smallpox by, for example, taking the scab of someone who had smallpox who was just recovering and grinding it up and sniffing it up their nose. And if you think about it, that's like vaccination. You're giving yourself a weakened or a small dose of a virus in the hope that your immune system will react and will learn how to protect you in case in the future you actually do get the virus and then maybe you don't get so sick. So this process had been going on for a long time, but Jenner figured out that you could inject people with an associated virus. It's not 100% clear what that virus was, and you would get a little infection, like from, let's say, cowpox or horsepox, so a virus from another species, and your immune system would react and it would protect you if you actually got smallpox. And it turned out that he was right. And this was based on an observation that women who were sort of milkmaids who'd been milking cows would get like a pox infection on their hands, but they wouldn't get very sick, but they never got smallpox. So vaccination was invented, in 1796, pretty much. And since then, many viruses and also bacteria we've developed vaccines for. And I think it's quite telling in a way that many people, let's say of my age or younger, let's say I'm 57, it's not a secret. But if you're, let's say, 40 or younger, you probably don't even know what half of these viruses that I'm listing on this slide even are, or what they did, because a lot of them are just gone, or they're no longer an issue. And the reason they're gone is because of vaccination. So vaccination is just, it's amazing what it does. Edward Jenner, a few years after he'd come up with this idea, and he tried vaccinating a small number of people, and this process had spread to different parts of the globe, he wrote a pamphlet, which you can read. It's a little challenging to read it. And in the pamphlet, I've highlighted a bit at the end, and he says, it now becomes too manifest to admit of controversy. So he's saying, there's no doubt anymore that the annihilation of the smallpox, the most dreadful scourge of the human species, must be the final result of this practice. So Jenner is saying 180 years in advance, the outcome of what I've invented, of what I've discovered, is that we're going to get rid of smallpox. And he was right. He was kind of lucky, but he was right. And it turns out it's more difficult with other viruses. But Jenner got it right. And there was a massive effort that culminated in about 1980 to rid the planet of smallpox. And by the way, the word vaccine has an interesting etymology because this was work that was done based on cows. Jenner was calling the cowpox virus, he was calling it Variolae vaccinae, which came from the Latin word for cows. So the reason we call it a vaccine is because he was working on cows, even though he actually thought that the virus was coming from horses, and it was like a horsepox infection. So that's perhaps interesting to people. Another thing which is of interest is that the anti-vax movement that is so sort of virulent today and so on, it's nothing new. It's been going on since before Jenner invented vaccination. So all of this stuff has been controversial for a very long time. And obviously, giving someone a virus is quite a questionable thing to do if you don't know what you're doing. And there was a lot of risk associated with inoculation and with early vaccination. But science progresses, and we learn a lot of things, and there's many different kinds of vaccines. And today, the vaccines are very clever, and they don't have the virus in them anymore. They've got a tiny little piece of it. You can't give someone a virus, with a vaccine like that. There have been issues, for example, with polio, where you have an attenuated or a weakened virus, and you give it to someone, you are giving them the virus, and things can go wrong. Okay, anyway, historically, this has been around for a long time. Okay, I'm getting to the end now. One of the questions that people sometimes ask is, they say, well, this all sounds very cool, and it's interesting, Vikings had smallpox and this was unknown, but what's the use of this work? Does it have any practical impact? And I think the best answer to that, or the best, the most obvious thing is that if you think about extinction, like these viruses are not around anymore, they've disappeared. If you think about extinction in the world of zoology, when an animal goes extinct, like a mammoth goes extinct, it's not coming back. And you're not going to have, for example, the birth of an elephant that's got a mammoth's head. Not even part of the animal is going to come back. But the situation with viruses is quite different. And a virus could, in theory, come back. And more likely is that a part of a virus could come back. And what we found in looking at ancient sequences, these ones actually are from hepatitis B, which is another virus that we've worked on. We found that the oldest sequences are different from the modern ones. And in this particular case that I'm highlighting, you can see in the bottom 3 sequences there's a gap in the DNA. There are 6 nucleotides that are not in the oldest sequences in this particular genotype. It's called genotype A, which is all over the world in hepatitis B, which, by the way, kills nearly 1 million people a year. And so you have this question, it's like, why did why did the genomes of the virus pick up those 6 nucleotides about 1,150 years ago, 1,500 years ago? And that mutation, or that, that inclusion of 6 nucleotides, it spread all over the world. What happened at that point in time? Did that change affect the fitness of the virus? Did it make it more dangerous? Did it replicate better? Did it transmit better? What happened? And the thing which is interesting is that that variation that we can see in the old viruses, it was invisible until we saw the ancient viruses. But we can now go and look at those ancient viruses and we can figure out what was happening there. And in particular, we could figure out if a modern virus were to lose those 6 nucleotides, what would happen? Would people still be protected by the vaccine or the antivirals, and would the diagnostic tools still work? So the thing which is interesting here is that all the viruses from the past, they sort of give us a library of variation that we can't see today. But once we see it in the past, we know that it could happen, that it would result in a virus that would work, and we can do tests on it. And it can prepare us for this kind of variation, which could happen again in the future. So there's quite a strong practical outcome from some of this work. Okay, and speaking of this work, everything that we've done, everything I've just talked about, it's been done by a large team of people who in some cases have worked for decades on this, and we have a very wide range of backgrounds. I'm actually a computer scientist to begin with. We have people who are working on human genomes. We have archaeologists. We have pox virus specialists. We have at least a couple of the best-known coronavirus virologists in the world, Ron Fouché and Christian Drosten on the team. Derek Smith is an influenza virologist who works on the design of the influenza vaccine for the WHO. And you have a large collection of people who have spent some years now working collaboratively to get this work done. So this is really a team effort. Okay, and the last slide here, for anyone who's interested, there's a bunch of very interesting books that you can read about smallpox. And I've just highlighted a couple of them. One I mentioned is by Donald Hopkins. That's on the top left. Another one which Jim, our host, is reading or has on his shelf to read is called Pox Americana. And then there's 2 at the bottom that I've put. One of them looks like a comic book. And in fact, it is a comic book a little bit, but it's very good. It's called How Pathogenic Viruses Think. And the one on the bottom right is also excellent by the same author, which is just an introduction to the immune system. So if you have an interest in those subjects, those bottom 2 books are quite thin, and they're also, the bottom left one about viruses is entertaining and quite easy. The bottom right one about the immune system, it's easily written, but the immune system is very complicated. And the top left one, I would highly encourage you to go and read some of the smallpox book, because the history is really incredible. And with that, I'm happy to take as many questions as we have time for.
Can you talk a little bit about how your training as a computer scientist kind of fits into this sort of research?
Aha, yes. I've turned off my screen sharing. It's very interesting. First of all, it's like a handicap, because I don't know anything about viruses, or at least when I started, I knew nothing about viruses. But at the same time, it's an advantage, because you can ask as many dumb questions as you want, and no one looks too surprised when you tell them that you don't know what a protein is or whatever. But the main thing, which is the value which gets added, is that once the DNA has come out of the sequencing machine, everything from there on is sort of a digital challenge, or a lot of the things from there on are digital challenges, where we have to take the fragmented DNA and we have to put it back together and try and find out what the virus sequence looks like, which in a case like this is actually very challenging, because we don't know what the original virus looked like. When we do that with a modern virus, we have sequences. We have modern sequences that we can compare the fragmented DNA to, and we can reconstruct the sequences quite well. In this case, we didn't have the old sequences, and it turns out they're quite different. So there's a lot of computational challenges in doing this. Another one, which is just sort of like basic engineering, is we process something like, I think, 125 billion reads from the sequencing machines, which is a lot of data, and it's a lot of work on high-performance computing clusters. And so computational knowledge, who would have guessed, but it actually comes in interesting. It's important and interesting in the biological context.
All right. So going back to one of the first pictures that you showed, the two boys side by side, one that hadn't been vaccinated, one that had. Can you talk a little bit about how the disease progresses when people caught smallpox?
I could. Just by the way, I've just switched myself onto participation mode. I can see the participants. We have some very knowledgeable pox virologists in the audience. I'm not sure if you're aware of that, Jim, or if other people on the call are aware of that, but we have some really, some people who really know about pox viruses. So I have to be a little bit careful. There's a variety of outcomes. It's similar in a way to the coronavirus that's going around at the moment. Some people have a very bad course, and you know, the disease affects them very badly and kills them. Some people have a mild infection. With smallpox, with modern smallpox, it's, you know, one way of thinking about it is there were two, or maybe some people would say 3 or 4, but let's say 2 different variants of the virus. One of them had a case fatality rate of less than 1%. So if you happen to get that virus, the probability of dying is very low, and you may have just a mild outcome. On the other hand, if you got the bad form of smallpox, you had a 30% chance of dying. And the progression is you have an incubation period that would last, I'm not sure how long, I think it's something like 10 days. And then you have all sorts of different symptoms that appear. Obviously, the most devastating one is the outbreak of the pox. I'm not sure if people know where the word pox comes from, or even why it's called smallpox, but there's other viruses that cause a rash, and in particular, the big pox was from gonorrhea, I believe, or syphilis. That was the big pox, and then they called it the smallpox, so it was called the smallpox. The virus could do different things. It could get into the eyes, for example. People would be turned blind. There's some quite specific differences from that were noted from about the year, let's say 900 or so onwards, where they learned to tell the difference between measles and smallpox based on some of the symptoms and the way that the disease progressed, and where did the rash appear and when, and so on.
You mentioned earlier that it's illegal to do experiments with smallpox. So there would be concern that smallpox, if it were to get out, could get into the wrong hands. Is that correct?
Yes.
And that's why you're limited in the kind of work that you can do with like these ancient strains of smallpox.
Yes, there's. I mean, you can't really do anything. There are people who do work on smallpox or on particular genes, for example, let's say at the CDC, but those things are done under very tightly controlled circumstances, and often, of course, work like that would be done on a similar pox virus, like, for example, vaccinia virus. And as I said, it might be done in a different species, first of all. So you might study what happens to a mouse if you knock out a particular gene or if you make a particular change. In some cases, you can do experiments on a virus where you can do things in a much safer way. But if you know, the rules that come from the WHO about working with pox virus, with smallpox virus genomes and material, is that you can't have more than, I think the number is still currently 500 nucleotides of DNA in an experiment or at any one time. So they're very restricted, and you have to ask for permission. So there's very few places in the world where anyone's doing any of this kind of work directly on anything that really is smallpox, but they operate on related viruses. And as for what's going on at the CDC or in Russia, there are occasional publications, but not very many.
All right. Dr. Terry Jones, thank you so much for joining us. Thank you all for being with us today. Thanks to the Museum's Liaison Committee for joining us. Please join us next week on October 21st at noon for the technology and science of looping, Cooking Beats with Kanika Cook, who's a local musician. You can register for next week's talk at smb.org. Each talk is free to attend and is open to the first 300 registrants. Thank you again for joining us today. Until next week, stay safe and stay curious.